The next type of quality assurance test is mutagenicity testing. Testing the preparation whether it is free from mutagenic agents or not. Testing the preparation whether it is mutagenic or not. It is performed by a test called as Ames test. Directly assessing potential carcinogens by testing for their ability to form tumors in animals is very difficult and expensive. Means to test tumor inducing ability of certain chemicals. It is difficult and expensive because several animals are required to be used for these types of carcinogenicity testings. Considering the fact that all the carcinogens are known to give positive mutagenicity test, that means all the carcinogens are mutagenic agents. We understand the definition of mutagen. So, a substance that causes mutation. Carcinogens means the substances that causes formation of carcinoma, that is cancer, which is also a result of genetic modification. Mutagens are also same. So carcinogens are mutagens and therefore they are known to give positive mutagenicity test. Bruce Ames described a very sensitive and simple bacterial test for detecting chemical mutagens. This Ames test it is used as a standard method for the detection of mutagenic activity of drugs and environmental chemicals. It is based on the use of specially constructed mutant strains of Salmonella typhimurium. This Salmonella typhimurium is an auxotropic mutant. It is auxotropic for histidine amino acid. That means it requires histidine for its growth. In absence of histidine, the organism is unable to grow. So these mutants are highly sensitive to the action of mutagenic agents and they get reverted to prototropes called as revertants. That means if these mutants are exposed to carcinogens or mutagenic agents, these mutants get a reversion to parental type. That means they get converted from auxotropic mutants to the revertants, that is of prototropic type. So this reversion indicates that the chemical is a mutagen. That is the principle of this Ames test. Now, this is the A part of this test in which we can see that the test compound, that test compound is mixed into the medium containing Salmonella typhimurium and then it is plated. So this is the medium. Now see, this is the medium that contains this test compound. So this test compound is added to this medium and this medium contains this auxotropic mutant. Remember that this Salmonella typhimurium, it is an auxotrophic mutant and therefore it will not grow in absence of histidine. Therefore, the medium which is used, it should contain some little concentration of histidine, that is just to support the growth. Unless and until there is growth, we cannot test the effect of this test compound on Salmonella typhimurium. So this medium must contain traces of histidine to support the growth. Then these plates are incubated, and after incubation we can see we may see these two types of results. First one is there may be development of large colonies. So large colonies are of prototrophs, that means revertants. This means that the test compound is a mutagen. That means there is reversion because the test compound is a mutagen. We may get this type of results: presence of large colonies of prototrophs and small colonies of auxotrophs. So these small colonies indicates that there is no mutation. However, we may find the presence of these large colonies. Then can we say that the test compound is mutagen? No. Compare this plate with this plate. What difference do you see? If a chemical is a mutagen, then the number of large colonies is more, and here the number of large colonies is very less. Then the reason why these large colonies appear. These colonies are the results of spontaneous mutation. So because of spontaneous mutation, which is not related to the action of chemical mutagen, so these large colonies may appear here. So this indicates that the test compound is not a mutagen. Another set is run parallel to it. This is for the test compound, and this is without the test compound, that is saline. So saline is mixed with the same plate containing Salmonella typhimurium with traces of histidine. It is incubated. You can see the presence of large colonies. So in absence of this test compound, also you can see presence of large colonies. So you can compare this plate with this plate. So this indicates that the test compound is not a mutagen. There are certain chemicals which when enter in our body, so these chemicals are processed through and a very vital organ in our body, that is liver. So liver enzymes acts on these chemicals and they convert these chemicals into different form, means these chemicals are transformed. So that is the function of liver, transformation of chemicals. That is one of the functions of liver. Detoxification of chemical compound, that is one of the functions of liver cells or hepatic cells. It may happen that chemicals which are non-mutagenic entering in our body, they are processed by these liver enzymes and they may get transformed into mutagenic substances. So non-mutagenic substances may get converted to mutagenic substances due to these liver enzymes. So to check the activation of such compounds, this compound is incubated with rat liver extract. That means it is incubated with the hepatic enzymes and then they are tested. Then the protocol is same again. Here we can see rat liver extract, it is incubated in presence of this test compound. So the if there is any transformation, it is allowed to occur, and then it is transferred onto the plate containing Salmonella typhimurium, which is grown on the medium containing traces of histidine. Then there will be a control without test compound, that is rat liver extract. It is processed in the similar manner and then it is incubated. After incubation again we'll see these two types of results. Presence of large colonies in large number indicates that the test compound is a mutagen. There may be presence of large colonies in very small number. Compare this plate with this control plate. Here we can see these large colonies are the results of spontaneous mutation, and this plate will also show the presence of small colonies of oxotroph. So this indicates that the test compound is not a mutagen. So this is how the efficiency of this test, it can be increased by the addition of this rat liver extract.